Good morning, students. All of you welcome for the second part of this cell. This is the revision session for the upcoming terminal exam. So, last lecture we have previously revised all about the cell. Now we'll discuss about the cell organelles. So, the first organelle that we are going to study is a plastid. Plastids are the cell organelles mainly present in the plant cell. There are three types of plastids mainly, which we are going to study in this topic is chloroplast, chromoplast and leucoplast. And somewhat historical background about the plastid is plastids is a membrane bound organelle found in cells of plant, algae and some eukaryotic organisms. So mainly it is found in the plant cell, but some algae and eukaryotic organisms also have the plastids, and this three different type of plastids have different functions. Plastids were discovered and named by Ernst Haeckel. Plastids are the site of manufacture and storage of important chemicals in cell. It is not only the thing that the chemicals are stored in the plastids. The chloroplast, which is mainly present in the green plant, is responsible for the synthesis of food by the process of photosynthesis. Photosynthesis is the process in which light is trapped by the light active chlorophyll by plants and food is produced. That is called as chloroplast, which is green in color, and the pigment present in the chloroplast is a chlorophyll, which is green in color, which entraps the sunlight and carry out the photosynthesis for the production of food. So please remember, the scientist who discovered the plastid is Ernst Haeckel, and plastid is present in plant, algae and eukaryotic organisms. Now actually we'll study the type of plastid, chloroplast. It contains chlorophyll. It is green color plastid. Green color is because of chlorophyll, and which is useful for photosynthesis for production of food. Second type of plastid is a leucoplast, which is nearly about transparent or whitish in color. The function of the leucoplast is storage of food or other compounds. Yes, it stores food or some other compounds like waste products which is needed to be excreted from the plant, so that is stored in the leucoplast. Third type of plastid is a chromoplast. It is present in fruit, flowers, ornamental plants, leaves, because ornamental plants leaves are colorful. So chromoplast gives color to the fruit, flower and leaves of the ornamental plant. There are more other type of plastids that I have represented in the slide. Okay, mainly you can see over here chromoplast, chloroplast and leucoplast, but still there are other type of plastids. So subtypes of leucoplast are amyloplast, which stores the starch. Elaioplast is a plastid which stores the lipids and fats. Okay, lipids and fats. Proteinoplast, it stores proteins. And the subtype of protoplastid is chloroplast and leucoplast, and the subtype of etioplast is chloroplast and chromoplast. So these are a type of plastids. Next cell organelle that we are going to study is endoplasmic reticulum. Now this is the unique cell organelle which is involved in the process of protein synthesis. Endoplasmic reticulum is a type of organelle made up of two subunits, rough endoplasmic reticulum and smooth endoplasmic reticulum. These are two types of endoplasmic reticulum. Endoplasmic reticulum is found in the most eukaryotic cells and forms network of flattened sac in rough endoplasmic reticulum and tubular structures in SER. Okay. Tubular structure is seen in SER and flattened sac-like structure is found in RER. Okay. Flattened and rough appearance is found in the rough endoplasmic reticulum. Rough because association of the ribosomes with the endoplasmic reticulum is rough endoplasmic reticulum. Means when ribosomes are associated with the endoplasmic reticulum, then it is called as rough endoplasmic reticulum. And the function of this rough endoplasmic reticulum is production of protein. And smooth endoplasmic reticulum, okay, which is a tubular structure which is involved in the lipid synthesis that I have mentioned in the next slide. The membranes of ER is continuous with nuclear membrane. Endoplasmic reticulum is discovered by scientist K.R. Porter. It is discovered by K.R. Porter. And as we have discussed, there are two types of endoplasmic reticulum, that is RER. Ribosomes are attached to endoplasmic reticulum, so it is called rough endoplasmic reticulum. It is involved in protein production. Smooth endoplasmic reticulum is involved in lipid synthesis. So let me tell you the location of the endoplasmic reticulums. Smooth endoplasmic reticulum is present near the cell membrane, which provides the lipids to the repair mechanism of the cell membrane. And rough endoplasmic reticulum is present near the nucleus, around the nucleus, and which provides protein for various purposes. This is the image of endoplasmic reticulum, and this is the transverse section of endoplasmic reticulum and nucleus. Okay, this is the nucleus, transverse section of nucleus, and this is the transverse sections of the endoplasmic reticulum. Now you can see the association of ribosomes with the endoplasmic reticulum. The blue spots you can see on the reticulum is nothing but the ribosome, and ribosomes are associated with the endoplasmic reticulum, means it is a rough endoplasmic reticulum. The foldings of endoplasmic reticulum is called as cisternae. This foldings you can see, these are the foldings of endoplasmic reticulum. They are called as cisternae. And the space inside you can see, inside the cisternae is a cisternal space. Means inside the endoplasmic reticulum the space is present. That space is cisternal space. Next cell organelle is Golgi apparatus. It is discovered by scientist Camillo Golgi. Camillo Golgi is the scientist, and depending upon his name, the name is given to the cell organelle is a Golgi apparatus. It is sac-like flattened structure and play important role in secretion of biomolecules and transportation of proteins. Secretion and transportation of biomolecule is done by Golgi apparatus. Okay, so for the transportation of biomolecule from one place to the another place inside the cell is done by the Golgi apparatus. Here you can see the image of Golgi apparatus. Golgi apparatus is also a flattened sac-like structures, and it is a bunch of sac-like structure parallel to each other, closely arranged, and as you can see, it produces the secretory vesicle. This is the secretory vesicle, and why this secretory vesicle is produced by the Golgi apparatus is proteins and other biomolecules like carbohydrates and lipids, vitamins, and other signaling molecules is enclosed in the secretory vesicle, and this vesicle is transported from one place to another place in the cell. So, transportation in the cell through cytoplasm is done. So, that is the function of the Golgi apparatus. Thank you. So, the rest of the revisions will do in the next lecture. Okay. So, if you don't understand or if you have further queries, you can call me and ask me, get clear your doubts.